Hello, my name is Chloe Brody, and my project is a study identifying zinc-containing neurons to widen research into zinc's role in neuroplasticity. So what is neuroplasticity? It's the ability of the brain to form and reorganize synaptic connections. In simpler terms, the ability of the brain to change itself. It typically happens in response to learning, experience, or injury, all scenarios where the brain will either develop new or find alternate pathways to perform a certain function or to get to a certain destination. Vesicular zinc is a pool of free-floating zinc in the brain that has been found to play a unique signaling function, leading to theories regarding its possible role as a neurotransmitter and that it plays a significant role in plasticity. There has been research done to show that this vesicular zinc is released from synapses in an activity-dependent manner, demonstrating its possible role in functional reorganization. ZNT3 is a protein that is responsible for the transportation of vesicular zinc, also known as synaptic zinc. There's a direct correlation between ZNT3 and synaptic zinc, meaning that the abundance of the protein is a signal of zinc levels. The protein is expressed in the brain and in the periphery. The brain structures that have the highest abundance of ZNT3 expression are associated with learning and memory, further supporting its role in plasticity. The most common current method to study the role of zinc in the brain is the germline ZNT3 knockout mouse model, in which the gene that encodes for the ZNT3 protein is deleted entirely, meaning that there is absolutely no zinc in the brain. It is used by observing the behavioral differences to those of a wild-type mouse that naturally contains and releases vesicular zinc. While the germline ZNT3 knockout model is effective, it has its limitations. The use of a conditional knockout mouse would increase the scope of what can be studied and unlock new experimental possibilities. Now, what is this conditional knockout mouse model? It allows for the manipulation of gene expression to allow for spatiotemporal study of a gene of interest, meaning that in the case of the conditional ZNT3 knockout model, researchers will be able to study the effects of zinc in the brain in specific brain structures and at certain times in a mouse's lifetime. There are steps that need to be taken in order to create and use this mouse model, and this is where my project comes into play. A Cre recombinase expression system needs to be utilized. Cre recombinase is an enzyme that causes genetic recombination, and a tamoxifen-induced Cre expression system is commonly used in conditional knockout models to achieve cell-type specific gene expression. The mouse studied in my project was the novel transgenic ZNT3 Cre rosa mouse that had the Cre AI9 expression system. This mouse has Cre recombinase genetically engineered into the ZNT3 locus, so therefore, Cre should be expressed in all ZNT3 expressing neurons. But we had to confirm that it actually is. With tamoxifen exposure, the Cre will be expressed and be able to bind to LOXP sites on the AI9 reporter construct and allow for the transcription of the red fluorescent protein, TD tomato. Localizing the fluorescence allows for the identification of zincergic neurons. The ZNT3 Cre rosa mouse will have both the Cre recombinase and AI9 reporter constructs. As seen here, the AI9 construct has a LOXP flanked stop sequence designed to prevent transcription of TD tomato unless Cre is present. This figure shows how TD tomato will be expressed in a Cre-dependent manner, and supposedly in all zincergic neurons, since Cre is only expressed in zincergic neurons. That brings us to the purpose. The goal of the study is to determine if and characterize a Cre recombinase expression system that can be used in the identification of zincergic neurons and the subsequent study of the role of zinc as a neurotransmitter and in neuroplasticity. It is to be noted that I did not participate in the breeding or the perfusions of the mice. The ZNT3 Cre and Cre-dependent AI9 reporter mice that were created by the Jackson Laboratory were bred to create the transgenic mouse with the Cre AI9 expression system used. The mice were administered an intraperitoneal injection of tamoxifen and left for a period of 4 weeks in controlled conditions to allow for TD tomato expression. Following euthanization, the mice were perfused. Their brains were removed and post-fixed overnight before being transferred to a cryoprotectant. Histological techniques were used to slice the brains into 40 micrometer thick frozen sagittal sections using a sliding microtome. The brain was cut with a steady force to minimize cutting artifacts. Once cut, tissue sections were transferred into wells filled with PBS on a storage tray. Immunofluorescence was used to amplify the fluorescent signal from the endogenous TD tomato expression. Anti-M cherry was the primary antibody used to recognize the target antigen, TD tomato. A biotinylated secondary antibody was used, followed by the tertiary antibody, streptavidin alexaflor 488. These antibodies were chosen specifically because they allow for maximum signal detection. Immunofluorescent labeling was also used to identify somatostatin and parvalbumin expressing inhibitory neurons. Tissue sections were mounted with PBS in order of location of the brain. Slides were left to dry before being cover-slipped with Floramount with DAPI. Then, fluorescence microscopy was used to view the sections. The Texas red and FITC green channels were used. The controls for this study were mice 4 and 6 because one of the two alleles was wild type for either Cre or AI9. Therefore, there was no Cre-mediated expression of TD tomato in either mouse. The image to the right supports this. There is no TD tomato fluorescent expression present. The following images show localization of ZNT3 expression in a lateral and medial section using in situ hybridization. These images dictate the expected labeling of ZNT3 neurons via Cre-dependent expression of TD tomato, which happen to be very different from this. Instead, there is Cre-dependent nonspecific expression in areas depending on the genotype. One of the possible explanations that arose was that the native TD tomato expression was too weak and needed to be amplified with the use of immunofluorescence techniques. The experimental mice with different combinations of the Cre-AI9 alleles were used to compare relative expression of TD tomato. As seen in the figures, the endogenous expression of TD tomato was minimal and localized to the ventral areas of the brain and lateral sections, and the olfactory areas and medial sections. Both images are from a mouse that was heterozygous for both alleles. These figures demonstrate the difference in Cre-dependent TD tomato expression among different genotypes. When analyzing the images above, it was seen that mouse 20, which was homozygous heterozygous for Cre-AI9, had a higher TD tomato expression intensity in the piriform area, part of the olfactory areas, than mouse 5, which was heterozygous homozygous. The immunofluorescence procedures did significantly amplify the TD tomato fluorescence signal to allow for more detailed visualization of the neurons. However, it did not reveal any new neurons as hoped. In the figure above, the image to the left was taken in the Texas red channel and shows endogenous TD tomato expression. The image to the right was taken in the FITC green channel and shows amplified TD tomato expression following the use of the antibodies. Immunofluorescence efficacy is demonstrated by the increased clarity of neuronal morphological structures like dendrites. There seemed to be minimal random labeling of a few neurons in the somatosensory and visual areas of the isocortex. More labeling was expected here, as these are fairly ZNT3 heavy areas. There was no labeling in the cerebellum, which was expected as there is fairly low expression of ZNT3 in the cerebellum. This image gives a comparison of the same area and the same mouse, but a lateral section on the left and a medial section on the right. When approaching the midline, it was seen that the TD tomato fluorescence decreased and that the labeled neurons followed the localization of the piriform area, a part of the olfactory cortical area. This mouse, mouse 20, exhibited the highest amplified and native TD tomato expression. However, it was still not what was expected when compared to the widespread ZNT3 expression with ISH as shown earlier. In an attempt to discover the reasoning behind the unexpected results, it was decided to dive deeper into the exploration of the neurons that were labeled. It was observed that the morphology closely resembled inhibitory neurons from their small cell body and lack of linear dendrites that relate to pyramidal shapes. Somatostatin and parvalbumin are markers of inhibitory neurons. Recent research has found that synaptic zinc plays a role in somatostatin-mediated inhibition, so it was decided to use double labeling to phenotype the cell types of the Cre-dependent nonspecific TD tomato expression. There was no labeling of somatostatin-expressing neurons. There was labeling of parvalbumin-expressing neurons. However, it was not co-localized with TD tomato, which was expected since parvalbumin has not been proven to have a relation to synaptic zinc. It's possible that these negative results were because of the antibodies, which may have been too old. The next steps are to repeat the somatostatin labeling with new antibodies to get more reliable results, since there still could be co-localization. This figure shows a composite of the merged images from the channels showing TD tomato and parvalbumin expression to demonstrate lack of signal co-localization. Parvalbumin expressing neurons are green and TD tomato expressing zincergic neurons are magenta. In conclusion, the preliminary results showed weak fluorescent signal, so immunofluorescence was used. This amplified the signal, but the expression was still unexpected as it was not as widespread as normal ZNT3 expression. The morphology of the labeled neurons resembled inhibitory neurons, so labeling of inhibitory markers was used to determine co-localization. There was no co-localization with parvalbumin. However, somatostatin labeling will be done in the near future. This study represents the start of the characterization process for a novel transgenic mouse. Once completed, this conditional knockout model will unlock several experimental possibilities for studying zinc's role in plasticity. Researchers will have the opportunity to study the role of zinc at certain times in a mouse's lifetime and in certain brain structures. Sophisticated cell analysis will be able to be performed with Cre-control tools like optogenetics. Optogenetics will allow for the use of light stimulation to spatiotemporally excite and inhibit neurons to control the activity of these zinc-containing neurons. The subset of the study that examines the correlation between the nonspecific Cre-dependent expression of TD tomato and inhibitory neurons will help to phenotype the neurons that were labeled and direct the focus of the research team to localize the flaws in the ZNT3 Cre model and understand the discrepancies between what was expected and what was observed. I would like to thank Dr. Richard Dick, Ashley Cho, the lab team, and the Department of Psychology at the University of Calgary for making this study possible and for their mentorship. I would also like to give a huge thank you to Dr. Garcia Diaz for her incredibly valuable guidance and feedback on my work throughout this year in preparation for the science fair.